Hello and welcome. This will be a recorded lecture on Chapter 12, a summary of the organization of the nervous system. The nervous system is divided into hierarchical categories, meaning from the overview, the large assemblages of functional units down to small subdivisions that explain all of the variation and depth of human behavior. The nervous system is our fast signaling system. Electrochemical impulses travel with the speed of light from one place to another, allowing us to have real-time sight and sound reactions. Now, looking at this first slide, we can see the breakdown, and there are 8 categories: the central nervous system and the peripheral nervous system. Central nervous system is the brain and spinal cord only. The peripheral nervous system are the peripheral nerves of the rest of the body. Under the peripheral nervous system, we notice 2 very different classifications of signaling nerves. There are those that are sensing the outside body and sending messages toward the central nervous system. That's toward the spinal cord and brain. Those are called the afferent or sensory nerves. The other functional unit comes after we've received all those sensations, compared them with our memories, to our knowledge, our skills, and we basically decide to do something. We send a message out to our so-called effectors. So muscles, glands, and fat can all be affected by this second efferent system. They are the motor nervous system. Those signals go away from the central nervous system. Now, that means it's important that when we see a nerve inside the body, such as a spinal nerve, it's most likely that there are sensory and motor nerves in that same packet, since we have to send both services to every part of the body. So there have to be sensory pathways going from the fingers toward the central nervous system, and motor commands coming back in order to affect a motor change. In the motor nervous system, we have the somatic system, which is all of our conscious and voluntary skeletal muscles, and the autonomic, those nerves which are beneath conscious control. Conscious control, subconscious control of things like cardiac muscle and the heartbeat, the smooth muscle, the muscles in the walls of our other organs. Glands, when do we secrete and when do we produce the secretion? Finally, adipose tissue, when do we store fat and when do we release it? Within the autonomic nervous system, we do see several situations where the various elements of the autonomic nervous system are organized around a specific whole body response. So sympathetic nerve stimulation is that kind of emergency response that's going to call for very high level muscular exertion. And parasympathetic is that which relaxes. It is called rest and digest because it's accompanied by a more calm body state, but the body is very busy inside doing something like digestion. Here is the neuron. We've met the neuron before. The characteristic of the neuron is branching. You see that on the body end of the cell. It has all the normal organelles, but those branching dendrites produce ends, which provide connections to surrounding cells. The long axon communicates the nerve impulse that originates in the body down to the other end, the telodendrion. You'll notice the branching at that end where there are multiple connections to the body of the next cell, the postsynaptic cell. So we're going to understand that a nerve is directional. It does run from one end, the body, where it can be stimulated and form an electrochemical impulse, down to the other end. So there is an upstream and downstream to nerve impulses. Taking a look further, you notice that that synaptic terminal is a space, and here's where our knowledge of the neuromuscular junction comes in handy. The only difference in this story and muscular contraction is that The postsynaptic membrane is not the fiber of a muscle, it's another nerve. So the elements are the same. The synapse that is between the presynaptic and postsynaptic membrane. The neurotransmitter substance shown here as green vesicles. Other than that, it's membranes, cytoplasm, and organelles, just like any other cell. We're going to look at synaptic function as well as cell type. The cell type that we just reviewed is the so-called multipolar neuron, here shown on the right. But there are three other types depending on where the nerves occur. The anaxonic neuron, you'll notice the only thing different about it is it has no long axon. This is where you are packing neurons together so closely that you don't expect to transmit the stimulation very far. You expect to distribute it out to hundreds or thousands, tens of thousands, or even in some places in the brain, hundreds of thousands of connections to surrounding cells. So the brain and sense organs have these anaxonic neurons. The bipolar neuron is shown with the cell body more or less in the middle of the axon, and sensory organs like sight, smell, and hearing do pack cells in this way for various reasons. The unipolar neuron is one that is similar, but the cell body is in the middle, but kind of offset from the main line of the axon, and we find this in the peripheral nervous system in sensory neurons. There are three kinds of neurons: sensory, the afferent system; motor, the efferent system, but sometimes it's not just two neurons, there are neurons in between, and those would be called interneurons or association neurons. What we think of as memory or recall or personality comes from, not from sensory signals directly, but from comparing those sensory signals to the memories, the knowledge, the experiences that we're having at the moment. And that's what we mean by an association. Three types of sensory receptors begin our story, and interoceptors are what are monitoring our internal systems. So when you talk about the digestion, respiration, heartbeat, urinary, and reproductive functions, you're talking about those autonomic systems that pretty much regulate themselves. Also, interoceptors are important in our internal senses, like taste, deep, pressure, and pain. Exteroceptors are those things that are sensing the external environment, either from afar. The external senses like touch, temperature, and pressure are located nearby in the skin, or at a distance. Sight, smell, and hearing are much, much more elaborate in the nerve endings. Specialized special sense organs are needed to interpret the source of environmental energy that is being detected and firing the nerves. Proprioceptors are a little more difficult to explain. They basically are packed into joints and skeletal muscles, so at all times they're stretching or contracting or rebounding, are monitoring the position and the movement of the body and reporting it on a constant basis to the balance organs and the brain, so that we always know the position our body is in. We do see a number of cells that are not neurons, called neuroglia. These are the support, the builders, the maintainers of our nervous systems. And we see them like ependymal cells, where they line passageways or cavities. We see astrocytes and oligodendrocytes, which basically hold neurons in place. And this is not so easy, because when you think about a neuron making synapses with another neuron, you can't just tack it down. There's a space, the synapse. So you have to hold this neuron and this neuron in such a position that that space is correct. If it's too broad or if it's too narrow, then the synapse does not work. So these positioning cells that hold these branching structures close to each other with the appropriate spacing are very important. Finally, microglia are phagocytes. Like white blood cells, they seek debris or waste or pathogen and basically keep the central nervous system very clean. We can see here a light micrograph, this showing from the central canal of the spinal cord, lined with those dark purple ependymal cells. Now the surrounding cells, that's gray matter. Gray matter are neurons, neurons, neurons packed together. But the ependymal cells line that canal so the cerebrospinal fluid is separated in its flow from the gray matter of the spinal cord. We see here in this drawing a number of these cells, and these cells are, the body of the cell is shown here in blue, the nucleus is present and obvious, and anywhere blue membrane is exposed, synapses can be formed. Whether these are cut ends from synapses that were there. Here are extensions that show the connection to this astrocyte, and these synapses are forming nerve connections. You can also see that these astrocytes can extend out, and notice how they form these plates. This is probably surrounding a capillary in the brain, and we'll explain why that's important in just a moment. These little hot dog buns that you see right here are the extended extended membranes of oligodendrocytes, and you can see how they mask a great deal of the blue membrane of the neuron, but at the same time provide a broad purchase on that flimsy hydrophobic membrane in order to hold it in place. Here's a better view of the oligodendrocyte on the left. Do you see how these cells extend their membranes and then wrap them around their axons? This oligodendrocyte has done the same thing here, but leaving a slight node exposed. Wherever the node is exposed, an electrochemical signal or a synapse can be propagated. But you'll notice here the myelin made of those membranes wraps around again and again to insulate that part which is covered. You see an oligodendrocyte here interacting with an astrocyte. You can see the spreading of the ends of those processes, in this case to hold an axon in place with plenty of exposed membrane. Over here, a very different story. The astrocytes have actually formed these broad plates which grow until they meet edge to edge. And when they meet edge to edge, they completely surround and control this blood vessel, a capillary, in the brain. Now, the reason that this is important is we're going to discover that inside the central nervous system, the brain and the spinal cord are not directly in contact with blood and blood plasma. There are blood vessels feeding the capillary beds in the brain, but you'll notice this astrocyte wrapping so that anything that comes out of the blood vessel or anything goes back in has to be regulated by this cell. The fluid that surrounds this outer space is not plasma or just interstitial fluid. It is a special fluid called cerebrospinal fluid, and is characteristic of our entire central nervous system. Something must hold it in and hold the blood plasma out. We're going to discover that in a moment. So here we see a typical peripheral nerve. This is the same kind of neuron we saw. The branches are sort of cut off by the figure. But here we have specialized nodes and internodes. And we notice this kind of alternating pattern. Each one of these little beads is a Schwann cell. And Schwann cells can wrap the axons of peripheral nerves, insulating these major portions of the axon and leaving only these nodes exposed. They do it by cell growth and cell division. Here's a Schwann cell which has approached an axon and just engulfed it, but once it gets to this point, one of these points begins to grow around and around and around, producing a multiple layer of membrane around the electrical active electrically active axon. The neuroglia also contain peripheral cells like satellite cells. These are sort of the building and grounds cells. In addition to these Schwann cells on the right, the satellite cells are there. They regulate oxygen and CO2, nutrient and neurotransmitter levels around the neurons in ganglia. This is necessary for the gray matter function that's outside the peripheral nervous system. When we think about routing these nerve bundles, they're going from the brain and the spine out into all parts of the periphery. So think about this left arm. I'm going to need sensory tracts that are coming from my left hand and toward the central nervous system. I'm going to need motor tracts to carry signals back to the muscles, glands, and fat about what to do in the particular moment. That means that it makes real sense when you're running nerves to bundle them, sort of like a wiring bundle in a car or the bundle of wires in a building. Schwann cells can accommodate that. You'll notice this drawing is almost unbelievable how a cell can surround. In this case, it looks like 8 or 9 axons. And wherever it contacts an axon, you get that circular growth around and around, around and around, separating the axon not only from the surrounding fluids and cerebrospinal fluid, leaving only the nodes exposed, but also separating them from one another. Electrical energy can pass from any conductor to any other conductor, and insulation is a common feature I think you're familiar with wires. The metal wire is surrounded by an insulating cover. Schwann cells can do that from the standpoint of a living cell. In the periphery of our body, an amazing thing happens that does not happen in the central nervous system. We can repair nerves. So here is a break in a nerve. You'll notice the fragmentation of Schwann cell. Here is the proximal stump, meaning over on this side, the nerve axle is still continuous with the nervous system to the left. But here it's been fragmented and cut off those downstream nerves from electrical signals. Basically, fragmenting the parts of the cell, producing damage and debris. When that damage and debris and the damage chemicals that result from the fragmenting of a nerve are established, macrophages, a type of phagocytic white blood cell, come around and clean up the area. Now, they'll not only engulf bacteria, they have a lot of debris, membrane, and cytoplasm to clean up. You can see that here. But you'll also notice that the Schwann cells have begun to divide and produce a bridge across this broken area. Once that bridge is complete, then the standard Schwann cell growth and differentiation commences, and eventually those new cells repair or link the pre-break and post-break neural material, wrap it in new nodes, and restore nervous function. Now, recall, though, that this occurs only in the periphery, where angiogenesis can produce new blood vessels and nerve growth can produce a restoration of sensation or, in some cases, motor control. Now, that does not happen, as you know, in the central nervous system. A stroke or a lesion in the spine producing paraplegic or quadriplegic condition is a permanent damage that does not grow back. In general, if it's in the cerebrospinal fluid, part of the central nervous system, it will not regenerate from damage. Now, to understand nerve signals, we have to think very deeply about the unit of function. You remember when we talked about muscles? What is it that really contracts? Well, we had to get clear down to that microscopic sarcomere to understand how a muscle contracts. We're going to find the same situation in nerves and nerve impulses. To understand how a cell can send an electrical signal, we're going to have to realize the electrical nature of all cells in the body. If you take a tiny voltmeter, you have to have pretty fine electrodes. You stick one electrode into the cytoplasm of the cell and lay a little flat plate electrode on the outside of that membrane. The voltmeter registers a voltage. That means each cell has a difference in concentration of positive and negative charges on the two sides of its membrane. It's like a little battery storing electrical energy, and this is called the transmembrane potential, or sometimes just the cell potential. Now, these are established by specific carriers that transport charged ions, things like sodium, potassium, and chloride, and establish these different concentrations. So I want to make this point. A living cell has a measurable voltage, and sometimes it varies from region of the body to region. This cell potential, measured in voltage, voltage is like electrical pressure. It measures the potential to do work of an electrical kind. The transmembrane potential is going to be established at rest. Number one, the resting potential. A cell which is not firing is going to be registering that voltage in a fairly constant way. Now, I say fairly constant. We will select an average reading to illustrate the transmission of nerve impulses. What I want to tell you is that as we move the body around, as there's compression and expansion, as there is flow and reduction in flow of blood to various parts of the body, as nutrition changes, as pH changes, there are perturbations to this resting potential. So if you want to think about it, think about this, minus 70 millivolts, and if you sat and watched a cell you were measuring, it would be going minus 69, minus 68, minus 70, minus 71, minus 72, kind of varying. You could basically see the needle wiggle back and forth. But basically it's varying around that resting average. Those variations, temporary and local and small magnitude, are caused by stimulus, by touch, by pressure, by chemistry, and they are constant inside the cells. But in essence, they don't amount to much. It's sort of like the ripples on the surface of a pond. They come and go according to conditions. However, if you produce a specific kind of stimulus, it reaches a threshold reading. That's called the action potential. This is an electrical impulse because it causes a change in the neuronal membrane. That membrane change is going to produce a tremendous change in the membrane all along the surface of the cell from axon to synapse. And once it gets established, once you cross the threshold, it's going to depolarize, meaning it's going to cause this change in electrical character from one end of the cell to the other. Once it reaches the telodendrion, the synapse, the arriving electrical impulse will stimulate the fusion of vesicles with the presynaptic membrane and the release of neurotransmitters into the synapse. You know that neurotransmitters bind receptors on the postsynaptic membrane, and if enough bind, it will generate an action potential in the next cell body, fire it, and the nerve impulse will race along. Now, sometimes these stimulations will arrive at a place with lots of gray matter that is linked in a very complex way. This pattern of electrical discharge in masses of neurons, such as the brain, the spinal cord, or the ganglia that have gray matter outside the central nervous system are signaling this final piece, the information processing piece. And that can be as simple as, have we delivered enough stimulation that the next nerve fires after neurotransmitter enters the synapse? Or it can be as complex as our ongoing sensing of our environment, decisions about what to do next, recall of our memories, expression of our personality, all those higher order functions. On the autonomic side, we have higher order functions such as sleep and such as psychological disease that relate to information processing. So here's our start up at the cell body where resting potential is established, and the flexing, the body movement, the chemistry, the pH are all contributing to variations that we call graded potentials. If those graded potentials add up to that minimum threshold value, and I want to emphasize that's a voltage value, they fire and the action potential races all over the body of the cell and down the axon to the other end. You can see that the teloid dendria split it up and deliver that impulse because it's, as I said, every part of the membrane of the cell. Deliver it to all the synapses. This is where the neurotransmitter and binding the postsynaptic membrane take place, and at the very simplest level, that neurotransmitter binding represents the simplest kind of information processing in the nervous system. Back to more, a little bit more basic electricity, because we do have the transmembrane potential. It results from physical factors. What are those factors that establish it and determine how it will vary? It begins with the extracellular fluid and the intracellular fluid of the cell itself. Intracellular fluid, the cytosol, is going to be different from the extracellular fluid, the non-living fluid that the cell lives in. There are going to be concentration gradients established for ions with charge, sodium and potassium major among those, but also involving charged entities like chloride and proteins. Because they are at rest, transported by carriers across the membrane in differing amounts. It is not an equilibrium. We typically will find much more sodium outside the cell than inside. We will typically find much more potassium inside than outside, and that is because of these specific membrane carriers. That establishes this electrical difference. The second thing is the selectively permeable membrane, and that's what we're talking about when we talk about these differences in carriers. Keep the right things in and the right things out. This membrane permeability varies based on the ion that is involved. So across the plasma membrane, we have gradients, chemical gradients, which establish that difference in concentration can establish a situation where diffusion can be an important force. You remember diffusion, if an ion can move, it will move from high concentration to low concentration. Electrical gradients are responding to the attractions and repulsions of charge. So positive and positive will repel each other. Positive and negative will attract each other. The potential difference, plus or minus, and the magnitude of the voltage reading is really a measure of how much force there is across that membrane. The higher the voltage, the more the potential difference, and that means the greater the capacity for work. Looking at a typical drawing, very simplified, of a cell, here across the center is the plasma membrane. So here we have the outside, here we have the inside. And what we've drawn here, up here I noticed they've drawn in this voltmeter, and you can see from 0 voltage, it measures both negative and positive potential difference. This resting cell is showing a minus 70 millivolt reading as the potential difference we call the resting potential or the transmembrane potential. This would be a nerve that's ready to fire, that can be stimulated to generate an electrical impulse. Now, the reason for this difference are these carriers shown one example each, but actually exist in many, many copies in the cells all over our body. Here, we are going to represent potassium with orange, so this protein carrier and this ion are both, this is potassium ion, and this is called a potassium leak channel. That means that it is open to potassium and only potassium. The transport is one way from inside to outside. No ATP is required. It's called a slow leak channel. Purple represents sodium, and here's the sodium carrier and the sodium ion, and it's a slow leak channel allowing sodium to move the opposite direction from outside to inside. The major player here is the sodium-potassium exchange pump. Notice that it uses ATP. Every time one ATP is consumed, 2 potassium are pumped into the cell and 3 sodium are pumped out. Now, the sodium-potassium pump is one that we used as our example of an active transport system at the beginning of the semester. If you recall, the sodium-potassium exchange pump accounts for 1/3 of the ATP we are burning when we are at rest. So 1/3 of our ATP just to run this pump and set up these transmembrane potentials. There are other players that are important to the millivolt readings that will register in the next few slides. And I want to call your attention to the negative chloride ions shown in green. They are mainly outside the cell. And these many, many purple globs and this row of minuses right inside, they represent the contents of the cell. The cell is filled with different kinds of proteins, and at physiological pH, proteins are generally negatively charged. So whereas protein and chloride contribute to the actual millivolt reading, they are not moved during this process. It is primarily the movement of sodium and potassium that's going to cause this needle to jump and show that electrical work is being done. Potential, again, represents the equilibrium at rest when the sodium-potassium pump is working and those slow leak channels are allowing the return of the ion to the opposite starting position. Now, equilibrium potential of a cell would result from the individual attractions and repulsions of the different ions. And we're going to look just quickly at sodium and potassium. If we look at a number of diagrams, I'm sorry that these are so busy as diagrams, we notice that at the resting stage, the resting potential of minus 70, that there is a strong chemical gradient. The diffusion at rest is a high force to push potassium out of the cell because it's very concentrated here, very dilute out here. So the diffusion force for moving out is very strong. However, the electrical gradient, since the outer part of the cell is much more positive, actually the potassium is being attracted by most of this negative charge. So the net force at rest for potassium movement is this minus this. It's a slight force to move potassium out, but not very strong. Down here in the lower left corner, very, very different. Here's a situation where diffusion is trying to would be happy to push sodium in. And also the electrical gradient is such that the negative charges inside are going to attract that sodium. So the net force for sodium movement at rest is very large. Now that means that at rest, what we're going to do, when we reach the action potential, we're going to pop open sodium gates, and sodium is going to rush in. Now when it rushes in, potassium is still staying in place except for the slow leak and the sodium-potassium pump. But as sodium rushes in, this millivolt reading, now that we've reached the action potential, is going to become more positive. The needle will swing to the right. That changes the relationship between outside and inside. And so it will reverse this relationship once the sodium moves across, and it is largely more positive here and more negative out here. Then the force for moving potassium out is higher, and the force for moving sodium in is lower. So at that point, we're going to close the sodium gates, open the potassium gates, and the potassium is going to rush out of the cell. It is the sequence of those gates opening that produces what we call depolarization, a change from minus 70 all the way to 0 and beyond. And then when the gates finally close and the pumps get back to work, establishing that resting potential with more sodium outside and more potassium inside, we're ready to fire again. Now, this all depends on a very complex array of channels, and it's important to realize the kind of channels and the kind of movement that are present in our membranes. There are those like this illustration. This would be exactly, in fact, what's being illustrated here is a neurotransmitter-like reaction. You'll notice that in the top illustration, that resting state, the two protein pieces are slammed shut and the ion is bouncing off. However, bind it with acetylcholine, that chemically gated channel pops open and the sodium, in this case, rushes in. A chemically gated channel, because a neurotransmitter-like binding to a receptor opens the channel. A voltage-gated channel is more complex. You'll notice it has two gates, and at minus 70, what we're going to do is the outer gate is closed and the inner gate is open. What happens is that we pop open that outer gate when we reach a threshold. Notice minus 70 is closed, but minus 68, 65, 62, no change. But if we reach minus 60, that's the threshold. That's the action potential. And that outer gate pops open. When that outer gate pops open, sodium rushes in. Now that's going to change the voltage, making it more positive. You can see in the bottom, it has swung all the way from minus 60 over to plus 30 millivolts. And when that occurs, you reach another threshold-like value where the inner gate swings closed and prevents movement of the ion. Now this is a mechanically gated channel. Just to be complete, there are some channels that are sort of like clothespins sitting there in the membrane, closed during a normal resting cell, but put a pressure on the membrane, a pressure on the tissue, and it literally kind of opens. And during that time when it's open, ions can cross over. So given those three kinds of channels, we have a chance to look now at the transmembrane potential across the plasma membrane and summarize some of its properties. There is a difference of chemical and ionic, of chemical and electrical concentrations that establishes that transmembrane potential. That's really what we mean by a selectively permeable membrane. The transmembrane potential is going to change in response to all the stimuli around it, the chemistry, the pH, the mechanical pressure, the movement. And that's going to change these chemical and physical conditions that we see. So let's take a look quickly at what we see as a membrane of a neuron functioning under graded potential. Up at the top, here is the resting state with carriers closed. They're just showing lots of sodium outside, and they're showing two carriers on opposite sides of the voltmeter in the center. The resting potential of minus 70 millivolts is shown. Now, if a membrane is exposed to a chemical condition that opens the sodium channels, you'll notice the sodium rushing in, and you'll also notice the millivolt reading changing. If those channels open only briefly, so it changes to minus 65, but then the pumps take it back to minus 70, then that would be an ongoing graded potential. But if enough rushes across that it fills sodium in this local area, then this sodium begins to spread out left and right from the points of entry. And that's where you can get a propagating change in this millivolt reading. You can see here from the point of entry, the initial stimulation, this is still minus 70 millivolt, but this intermediate position is minus 65, and it is increasing towards zero. Here we've reached minus 60 millivolts, which we will use later as a typical value for the action potential. So let's think a little bit about graded potentials. Graded potentials can occur on every membrane all over the body, every type of cell, whether it's sensitive or not. Graded potentials can be depolarizing, meaning they're moving from minus 70 towards zero, or hyperpolarizing. They're moving from minus 70 to minus 75 and further away from zero. There are four basic characteristics of these graded potentials. It's most charged at the site where you stimulate it, whether it's a chemical arriving or an electrical impulse that's arriving at a synapse. It could be a mechanical disturbance where you've been poked at the membrane in that particular place. Get further and further away from that site of stimulation, and the graded potential diminishes. The effect spreads passively, and basically they die out. If it's a true graded potential, it's sort of like throwing that stone into a calm pond, you know, the ring that starts out, and it's highest right near where the stone hits. But as it spreads out, as that line gets bigger and bigger, the wave becomes smaller and smaller and eventually disappears. That's sort of a good model for thinking about graded potentials. They are produced in a particular place, and they die out. Now, this graded change is going on everywhere, and the different graded potentials on different parts of the cells are interacting with each other at all times. But I want to emphasize, the stronger the stimulus, meaning the more synapses that are stimulated with electrical impulses, the more chemical that's delivered to a certain place, the stronger the poke to the membrane, then the larger and larger the area that is affected. So when we look at a electrical chart, down the bottom it's just time, and over on the left we see millivolt readings, and we're starting out at a minus 70 right here. This is the minus 70 resting potential, and we apply a chemical stimulus. We're depolarizing because polar just means different. So as you become more different, you are polarizing. As you become less different, as you go towards zero, you are depolarizing. Remove the chemical stimulus, and you will see the pumps get busy and reestablish the resting potential. Here we have a different chemical applied, and it will hyperpolarize. It takes it to minus 80 before you relieve it and return it to minus 70 again. These are graded potentials. They're local, they're short-term, and they are of small effect. Also notice they do not reach an action potential trigger. Now, the action potential is going to be very different. And this is where I'm going to start my next recorded lecture. So goodbye, and see you in the next presentation.